few words. As Gabriella, we walked quite a bit around Prague. It's a beautiful, beautiful city. So it's wonderful to be here. I've really enjoyed it. As I'm talking, if there are any questions, please just stop me. All right, I do not mind being interrupted. So if there's anything I say that it doesn't make sense, just stop and ask, and I will be very happy to explain it. So this is where I come from. Knoxville, Tennessee is right there. It's the University of Tennessee. I am in the Center for Environmental Biotechnology. This is our campus, and we have about 27,000 students from over 100 different countries. If anyone is ever interested in, as a student, if anyone is ever interested in doing internships within the university, please ask me. We're always looking for international internship students, and they're usually very hard to find because students never want to leave their home. But if anybody ever wants to leave and do some research for 6 months or a year, we can always try to find a fellowship somewhere. Right now it's very difficult just because the economy is very difficult, but we try. So we would love to have students visit. So I'll give you a little lesson on bioimaging this morning. Bioimaging or biomedical imaging is something for you younger students to keep an eye on, because at some point in your later life when you go to the doctor, things are going to be very different. And we're moving into personalized medicine. We are going to genome sequence you and see what genes are off inside of you before you ever even go to a doctor. All right, so we'll be able to predict what diseases you might encounter later in your life, and we will scan you for those diseases using things like bioimaging technology. All right, right now this is all very preliminary work on our part. This is over about the last 3 years we have been doing this bioimaging. So it's new, but it's up and coming. It's a very fast moving field. So the bioimaging is the visual representation, characterization, and quantification of biologic processes within living intact organisms. Why would you want to do that? For example, you can inject a virus into a mouse, into a living mouse as it's running around, and watch how that virus infects that mouse. What cells does it enter? You can expose that mouse to a carcinogen and watch tumor formation happening. I mean, right there on a camera, you can watch a tumor being formed. So you know exactly what cells are being affected with that carcinogen. So it has huge medical implications when you can watch a living organism respond to something. And in the old days, what would you do? You would expose your mouse to a chemical. You would expose 100 mice to a chemical, and every day you would kill a mouse, dissect it, and look at it and see what was going on. The next day you would dissect your next mouse. It was huge. It was extremely costly. Took so much time, and you learned very little from doing that. This bioimaging technology changes all that. And you can see some of these images, for example, here, that resets. If you're watching that, you can see that that mouse was injected with tumor cells, and you can see that tumor formation happening. It's sped up, but you can see that happening right before your eyes within a living mouse. And there's lots of imaging machinery out there to do this type of bioimaging. And some of these should be very familiar to you. So you have things like positron emission tomography, single photon emission computed tomography, PET and SPECT scans. We just reduce them in size so we can look at mice instead of humans. So we have micro PETs and micro SPECTs. So put a radio tracer inside your mouse, stick your mouse in the micro PET machine, and watch what that radio tracer does within the mouse. Computed tomography imaging, a micro CT instrument. Take X-ray slices of your mouse, and then the computer reassembles those slices into a 3D image. The MRI, which most of you probably are familiar with and probably have been inside of one at some point. Put your mouse in there and use a very ultra-strong magnet to rearrange the nuclei to get a 3D representation of the mouse. What I'll be focusing on is optical imaging, which is a very basic camera system that will peer inside of a living animal and look for light. And that light is either bioluminescent light or fluorescent light. Hopefully this starts. It's a little stuck, but we'll. So this is the machine. So your mouse or other small animal, at this point it only works with small animals. We can't put a human inside the machine because we can't see very far inside a human yet with our current camera technology. But we can put a little mouse in there. We have an anesthesia manifold that knocks your mouse out so it doesn't move around while you're trying to image. We have a filter wheel up here that holds any nanometer filter, so any fluorescent imaging that you want to do at any wavelength works just fine in the system. And then on the top up there we have an EMCCD camera, which is an electron multiplying cooled charged device camera, a minus 70 to minus 80 degree cooled camera, ultra sensitive, very little background photon counts in this camera system. And the EM, the electron multiplying capability, makes its frame capture rate extremely fast. So it takes pictures so fast that we no longer have to anesthetize animals, which is really, really incredible. So I wish this would go. It's stuck. But anyway, if you saw this image, this little mouse is running around in the chamber. And we're watching the tumor as the mouse moves. The camera keeps up with it. So the tumor, little glowing tumor, moves with the mouse. So the whole point is no anesthetic. Put your living mouse inside the chamber, image it as it's running around. Why is that a big deal? Because we always have to add anesthetic. Almost to any animal studies we do include anesthetic. We don't want to add another chemical to our experiment like an anesthetic agent. We don't know what the anesthesia really does, how it interferes with our experiment. So if we can leave it out, all the better. So here's your bioluminescent imaging light. Right now we use the firefly luciferase gene. We take the gene from the firefly, the light gene, and we put it into anything we want. All right, so we can take our firefly luciferase gene, hook it to some promoter that turns on whatever gene we want turned on inside that mouse, inject it into the mouse. When you use firefly luciferase, you have to add a substrate called luciferin. So once you have your firefly luciferase gene inside the mouse, then you have to add this luciferin substrate every time you want to see a light signal. So add luciferin to the mouse. The luciferin dissipates throughout the mouse's body, finds the cells that you implanted within the mouse. Those cells produce a burst of bioluminescent light, and you can see that light through the skin of the mouse. Your other alternative way to do optical imaging is fluorescence. And the most common fluorescent protein is green fluorescent protein. Now there's red fluorescent protein, yellow fluorescent protein, infrared fluorescent proteins. There's any color of fluorescent protein you can imagine can be used. So with fluorescent proteins, do the same thing. Implant your fluorescent cell inside the mouse. The machine will produce an excitation wavelength, which will excite the fluorescent protein. And that protein then produces light. So you see a fluorescent signal coming out of the mouse. All right, and there are problems with both systems. With bioluminescence, the way it's currently done with the firefly luciferase gene is that it's extremely invasive. You have to implant the cells in the mouse, and then every single time that you want to see light, you have to inject luciferin. All right, so that's needle stick after needle stick after needle stick. Your mouse gets extremely angry. It gets very stressed, and you do not want that to occur during an experiment. It's very expensive. That luciferin substrate is, for us, it's $300 per gram. It's very unstable. Once you take it out of the refrigerator and use it, you throw the rest away. So it's a very expensive technique to do, and lots of hands-on work. The needle, the mouse, constantly injecting the mouse, so it just takes lots of technician time. Fluorescent imaging, the big problem is autofluorescence. This mouse is under bioluminescent imaging. There's no background. We see exactly where the cells are. There are the cells, there are the cells. No background whatsoever. You do not you bioluminesce a little bit, but nothing that a camera can detect. So essentially, living things do not bioluminesce. Almost all living things fluoresce. So if we put you under the camera, you would look just like the mouse. Your entire body would glow. So in this case, the cells here are very easy to see because there are lots of cells stuck in the mouse right at that position. But imagine if you had a tiny, tiny spot of cells, you would never see it within this huge background fluorescence of the mouse tissue. And that's the biggest problem with fluorescent imaging. And they're trying to go into infrared colors, which has less background. They're doing all sorts of computer algorithms to get rid of that background fluorescence. So it's a huge problem that they're probably going to solve. They're working on it. So what we do is make, 20 years ago, when we first started all this, we made bioluminescent bioreporters. And instead of a firefly luciferase gene, we worked with the bacterial luciferase gene. And at some point, we figured, why not try to get the bacterial luciferase gene to work in a eukaryotic cell? So go from prokaryotic genetic system to a eukaryotic genetic system. And why would we want to do that? Because the bacterial luciferase does not require that luciferin substrate. All right, and that gives us a huge advantage. So now if we could make bacterial luciferase work inside of a mouse, we no longer have to add that luciferin substrate. So put your cells in the mouse, you are done. Put your mouse in the imaging chamber, you don't have to add anything. The cells make the light themselves. And they do that because of this reaction. So the bacterial luciferase contains the genes LuxC, D, A, B, and E, run by a single promoter off the Lux operon. The LuxAB genes come together to form the LuxAB protein. And that LuxAB protein is exactly like the firefly luciferase protein, except with firefly luciferase, this is all you get. Okay, at this point, you're done. When you want this to glow, to bioluminesce, you add a syringe full of luciferin. And then that protein will produce light. But with bacterial luciferase, you have this other component of the reaction, the LuxD, the LuxC, and the LuxE genes, which makes up this protein complex, which converts myristic acid, which comes from lipids that were supposed to go to the membrane biosynthesis. Instead, some are diverted to make myristic acid. It converts the myristic acid into a myristoaldehyde. That myristoaldehyde is essentially the luciferin substrate. So you can take that luciferin aldehyde, and there's also an FRP gene, which makes, I'll go through NADPH and NADPH2 later. But that myristoaldehyde then is what drives the bioluminescent reaction. So this is essentially the luciferin that we inject to make the luciferase bioluminesce. Firefly luciferase, you have to inject it. Bacterial luciferase, it's all there. The cell does everything. And some of the posters over there work with these bacterial bioluminescent bioreporters. So we can take these bacterial luciferase genes and inject them into bacteria and make them bioluminesce in response to usually chemical contaminants. Okay, like mercury, for example, as we have a poster over there. All right, so take these out in the environment. When they glow, when they bioluminesce, we know that mercury is present. We need to go out and clean up that site. And there are lots of other uses for these bacterial bioluminescent bioreporters. So now we're going to switch it over to, we're going to take those bacterial luciferase genes and make them eukaryotic. And that's not easy. Okay, so this started about 10 years ago when we tried to get these bacterial luciferase genes to be eukaryotic rather than prokaryotic. And there's several hurdles you have to jump over to make this work. Number one being that certain codons are liked better in prokaryotes rather than eukaryotes. And nowadays you have computer programs, and you can take your prokaryotic gene and put it in the computer program, and it will tell you if it will work in a eukaryotic organism. and it will tell you what codons, what 3 base pairs should be switched to make them more eukaryotic likable. Okay? So for example, here's the LuxE gene, wild type, that we pulled out of the bacterial cell. P is its probability of functioning, of making a functional protein in a eukaryotic system. For the LuxE gene, it had about a 44% chance of making a functional protein in a eukaryote. All right, so that protein would not have worked. We would not have gotten a functioning LuxE protein. Our bacterial luciferase reaction never would have worked in a eukaryotic organism the way it was originally set up. So we can go through now and make a LuxE optimized. So we can run it through a computer program, change every single codon from prokaryotic to eukaryotic without changing the amino acid sequence, because we have lots of different codons coding for the same amino acid, so we can switch those things around. No problem whatsoever. So in our LuxE optimized gene, once we codon optimized it, it has a 99.9% chance of making a protein that a eukaryote could read. Okay, so it will work in a eukaryote. And we did that throughout these holes. So you have 88% chance, 99, 99, 99, 99, for all 5 genes of this Lux operon. So they're going to make functional proteins. Whether they all work together, we don't know. But at least we fixed the genetic sequence to make it less prokaryotic, more eukaryotic. All right, and then the other big problem with prokaryotes versus eukaryotes, prokaryotes get to do this. Take one promoter and run several genes off of one promoter. So prokaryotes work with an operon. Eukaryotes do not. All right, so eukaryotes, it's typically one promoter, one gene. One promoter, one gene. One promoter, one gene. And your genome runs along just like that. So we have to trick our eukaryotic system into driving several genes off of a single promoter. And we do that through adding these 2A elements. And 2A elements are genetic elements that come from viruses. Viruses can do anything. All right, they're the most remarkable living entity we've ever worked with. Double-stranded DNA genomes, double-stranded RNA genomes, single-stranded DNA genomes, single-stranded RNA genomes. It's just amazing what viruses can all do. So it's not a surprise that this 2A element comes from a virus. But the 2A element links all of our genes together. So now we're going to have a LuxA gene, 2A element, followed by the LuxB gene, 2A element, followed by the LuxC gene, and so on. So it's 5 genes, but they look like one within a eukaryotic system. So we're going to make not a protein, we're going to make a polyprotein. We're going to make 5 proteins off of one promoter, and then these 2A elements let us snap those proteins apart once we're done making them. Okay, so one promoter making one protein, which is actually 5 proteins, that later get just broken apart into 5 different proteins. Okay, so that solves the problem of one gene, one promoter that we have to fix when going from a bacterial genome to a eukaryotic genome. And lastly, this FMNH2, if you remember back to the equation, FMNH2 is the gas, the gasoline that keeps the reaction going. Bacteria have lots of FMNH2. Eukaryotic cells do not have those huge supplies of FMNH2. So we found another gene called FRP, which we added to our Lux operon to make this, to convert the FMN to the FMNH2. So now we have a big gas tank inside the cell. That gas tank drives the bioluminescent reaction. So once we had all this in place, we were able to force those eukaryotic cells to make bioluminescent light entirely on their own. Okay, so no more substrate needs to be added. And that's the sequence. So there's one promoter, and you choose the promoter. So you can, I'll go through some examples in a second, but pick whatever promoter you want and link it to your bioluminescent genes. So there's your LuxC with a 2A linker, hooking it to LuxD, LuxA, LuxB, LuxE, FRP. So throw it in a eukaryotic cell, make a polyprotein, break off all those individual proteins, and you have the complete bacterial lux system within a eukaryotic cell. With the 2A, it's something with the ribosome. I'm not sure it's completely understood in full, but some of that ribosome gets kinked up within that 2A sequence, and it snaps the protein. It's remarkable how it works.
So it's actually produced as individual proteins?
No, it's produced as a polyprotein. And then later it gets snapped apart as it goes through the translation process. Okay, and nowadays, so this took a long time for us to build. We are now optimizing this even more. And when we want to optimize this now, we just go on the computer and type out an ATGC DNA sequence and email it to a place like GeneArt, that is a synthetic biology company. And they, in a few weeks, it usually takes 2 weeks, they just send us back this vector. All right, there's nothing to it. And it's remarkable with synthetic biology what you can do nowadays. And for all of the older people in here who ever did molecular biology and had to cut and paste genes and restriction sites and run GELDs, all that stuff is history, because you just order your genes, your vectors, your plasmids, they show up in a tube and you're done. So do everything on the computer nowadays. So it's really remarkable what synthetic biology is doing. So what benefit does having a bacterial luciferase function in a eukaryotic organism do for us? Gives us huge amounts of data. All right, so here is a HEK293 cell, human embryonic kidney cell containing our bacterial luciferase genes. We're monitoring light here, bioluminescent light in photons per second over time. We're up to 84 hours. The blue line is an HEK293 cell containing bacterial luciferase. We put the genes in the cell, and that's all we do. We put the cell in a machine that reads bioluminescent light. You can see these 100 or so points on the graph, and the line of that graph, and just the red is just a HEK cell without bioluminescence in it. Okay, so tons of data. If you're working with firefly luciferase, your experiment would look like this. All right, you'd get a graph with 5 points. And how do you make this graph? You take your bunch of mice at 6 hours, you pull them out of the cage, you inject them with luciferin substrate, you put them in the imaging chamber and take your picture. And that gives you your 6-hour time point. You then put your mouse, you can't constantly be putting luciferin. Luciferin is toxic in and of itself. You can't constantly put luciferin inside the mouse or it will die. So you have to give the mice some time to rest. So take the mice, put them back in their cage, come back later at your 24-hour time point, take your mice out of the cage, inject them with luciferin, put them in the imaging chamber, take your picture. And there's point number 2, and point number 3, point number 4, point number 5. All right, so if I wanted to monitor some chemical that could be toxic to humans, would I rather have that graph with 5 points telling me what's occurring, or that graph with 100 points telling me exactly what's occurring? And that's what we're trying to sell this technology on, is that it's a huge amount of data. All right, there's no more intermittent time points. It's a constant watching of bioluminescence within cells. So here, for example, we can do toxicity profiling. We're going to make the cells, we made these HEK cells, bioluminesce all the time. They are constitutively bioluminescent. And we put them in a 24-well microtiter plate. So you see 3 welds within the microtiter plate. Here's our positive control. They make light. They're always making light in this scenario. We're going to add zeaxanthin as our toxic chemical. Zeaxanthin is just an antibiotic DNA intercalator, so it's going to kill the cells. So at 100 micrograms per mil zeaxanthin, you can see our red hot spots, which were lots of bioluminescence, are gone. Okay, so those cells are hurting. We are harming those cells. They're losing their bioluminescent light, and all the way down. 200 micrograms less light, 300 micrograms even less. 400 micrograms, we're down to this light blue. Very little bioluminescent light being made. And this is just a control with the media showing that there's no background bioluminescence with bioluminescent light. And if you'd graph out those time points, so here's our zeolite concentration, 0, 100, 200, 300, 400, versus our bioluminescent output in photons per second. So at 0 micrograms per mil, zeolite is our highest light level. So they're constitutively bioluminescent, always glowing. Add a little bit of zeolite, light reduces, a little bit more, reduces more, a little bit more, reduces more. So nice straight line graphs just tells us that it's a dose-dependent response. All right, so we know the exact dose we're hitting those cells with based on the simple measurement of bioluminescent light. And that would be the graph you would get. So that's the total experiment. Can't remember how many data points. I think it's about 97 to 100 data points within that graph. And the only reason we even have data points is because our camera is an older system that just takes longer to take pictures. But if we had a really fast, if we had one of the newer, faster cameras, it would be an unlimited number of data points. You wouldn't even put points on the graph. It would just be a line of massive data points. Okay, so that's our graph of the whole experiment. And the benefit that gives us is it tells us exactly when things are happening. All right, we don't have 2 points that we connect with a line over 24 hours and say, within this 24-hour period, the 300 and 400 microgram samples had an effect. All right, that does not tell you much. It just says, within this range of hours, something happened. Here we got it right down. So at 3.25 hours, the 300 and 400 microgram per mil zeolite concentrations showed significant toxicity. At 8.25 hours, the 200 microgram per mil zeolite concentration showed significant toxicity. And at 9.5 hours, the 100, everything was significantly toxic. Okay, so it gives us those exact data points as to when things are occurring. So we did these experiments side by side. Lux, bacterial luciferase, Luke, firefly luciferase assay. So time points assayed, we did 97 points in that graph. We did 5 with firefly luciferase, just because you always have to add substrate. We had to prepare 15 samples within a 24-well microtiter plate. That was triplicate samples, times 5. For firefly luciferase, it was 75 samples, because once you add the luciferin to the microtiter plate well, that experiment is finished, because you have killed those cells, so you have to have multiple welds set up for every time point you want to do. Reagent costs, once again, no substrate, so it costs us nothing in substrate to do these experiments. About $100 US to buy the luciferin. Hands-on time took us 20 minutes to set up our experiment. Took an hour to do it the old way. And down here, sample destruction. No, we do not have to kill our cells. We can just watch them continuously. Firefly luciferase, you must kill your cells. Now you can switch this around too. So rather than having constitutive cells that glow all the time, you can make your cells dark and have them glow in response to a chemical. So in this case, we're not looking at how toxic a chemical is to a cell. We're going to detect something that's toxic. Okay, so our promoter is a tet-on promoter. It's a promoter that detects tetracycline, antibiotic tetracycline. Doxycycline is the other name for it. So we have a eukaryotic bioreporter for doxycycline. All right, so if your body encounters a toxic chemical or something even worse like something that's a signature carcinogen, we could make your organ, for example, glow. All right, so you go to the doctor and you get scanned, and if we see your organ glowing, then we know something's not right, and we need to take a closer look. Perhaps it's the beginning of cancer. Okay, so it'd be an easy way to detect, preclinical imaging for disease states. So here, for example, we're going to make the bioreporter detect doxycycline. And once again, lots of data points. And instead of being high light levels initially, it is now dark, no light being produced. We add doxycycline to the experiment. And as the doxycycline concentration builds up over time, we get more and more light being produced from our cells. Just an example of the steady state of bioluminescence. For our system, when you add the cells to the mouse and start imaging them, you get pretty much a straight line of photon per second counts coming out of that mouse. If you do it with firefly luciferase, you'll get more light being produced. Okay, so you can see it's higher light levels in that same spot, a bigger light signal coming out of that mouse. But it's higher but shorter in length because you're adding that luciferin substrate. So put the luciferin in the mouse, it dissipates throughout the mouse. It hits those cells make a burst of light, luciferin goes away. Okay, so you get high light, okay, 10 to the 8th compared to ours, which is 10 to the 5th, but it dissipates rapidly. And once again, just a comparison between the two, bacterial luciferase lux versus firefly luciferase luc. You can tell, you can do your cell counts just by the imaging, what we call the region of imaging within that mouse. So your computer will find these imaging areas on the mouse automatically and give you a photon per count number, which really doesn't mean anything. It's all part of the computer system that they use for this bioimaging. But you can count, so we have 500,000 cells in that spot. We put 500,000 cells in this spot, and based on our light reaction and our calculation, it comes to about 500,000 cells detectable with our light response. So you can do actual cell counts inside of the mouse, and that's very important for toxicity testing because you want to know if I inject a toxic chemical or, better yet, a drug that should treat cancer. I can put tumor cells inside of you, add the drug, give you the drug, and see how well those tumor cells respond to that drug. Okay, and ultimately, we don't want to do that with you. We want to do that in microtiter plates. So when you go to the doctor with cancer, for example, the way we treat cancer is to give you the most popular drug. You know, we don't quite know how well it's going to work. We're going to give it to you, have you try it. If you have bad side effects, we're going to switch it. If it has no effect, we're going to give you the next drug. Okay, so you really become the guinea pig if you have something like cancer, something like the more uncommon cancers that we don't quite understand yet. All right, so what we want to do is pull the cells out of you, pull your tumor cells out of you, put them in a microtiter plate, make them bioluminescent by inserting our genes, and then we can add whatever drugs we want to that microtiter plate. Whatever drug works best, we call you back to the doctor and say, here's the drug you get. It had no side effects on other cells of yours that we put in the microtiter plate, and it killed your tumor cells. So here you go. This is the best drug we can give you from day one, rather than trial and error to see what works best. All right, so you saw mice throughout this presentation. We're getting rid of mice. All right, the Europeans have the REACH directive, where all these chemicals now have to be tested for toxicity. And someone said, if we're going to test millions of drugs for toxicity, we do that with mice, we are going to be killing billions of mice. And someone said, ooh, that's not good. So let's make everybody test all these chemicals, and let's make them do it without animals, which is impossible. All right, but it's the REACH directive. The U.S. has its own directive to follow REACH. We have to do it. So we have to use fewer animals in research. And one of the answers to using fewer animals is to substitute mice with something less cute. Okay? So the zebrafish is what we use. The zebrafish is going to be one of the big replacements for mice. You know, the zebrafish are very inexpensive to maintain. You just need a few fish tanks, and you can have thousands of zebrafish. We have sequenced the genome of the zebrafish. UI and the zebrafish are almost identical. We always say how close the mouse is to us, but the zebrafish is even closer. So it's a perfect substitute model for the mouse. All right, so we'll use the zebrafish in replace of mice until the public starts getting angry that we're using zebrafish. But we had to do something to stop using all the mice that we use in research. So we are trying to also, besides make the mice bioluminescent, make the zebrafish bioluminescent. And we have some of the Lux genes working in the zebrafish. This is very much research in progress. So you can see 3 little zebrafish swimming around down here that we imaged. And this guy is bioluminescing. All right, so if we can get this to work in zebrafish, it's another huge step towards toxicity monitoring, drug monitoring. And this is even for environmental monitoring. You could put zebrafish out in the river in a cage, and if you saw the zebrafish glowing, you would know that something toxic was coming through your water, and you need to go look what it was. For aquarium users, the big problem with aquariums is the buildup of ammonia, and we have plans to make an ammonia-sensing zebrafish. So you could put that in your fish tank. When you go to bed at night and you turn off the lights and your zebrafish is glowing, it means you better clean your tank tomorrow before the ammonia builds up and kills the rest of your fish. And that would probably make more money than any biomedical zebrafish applications we could think of, which is really sad, but that's how it goes. So we're getting rid of mice. We're going to zebrafish. After zebrafish, we are going to do the human on a chip. So we also want to make your organs bioluminescing on a microfluidic chip. And I don't know how the funding is over here for the human on a chip project, but for the U.S., the Department of Defense is pouring billions of dollars into the human on a chip project. So then we aren't using any animals. We're going to grow your organs on a microfluidic chip in a substitute blood supply. And then we will inject, for example, the cancer drug into your human body on a chip, and that cancer drug will go to your liver and be processed. If it's broken down into other metabolites that might be toxic, that then goes to your other organs. So we'll have your heart on a chip, your lungs on a chip, every organ on a chip. And that's what it's coming to. And that's just to get rid of all animal use in research. And that's definitely coming. There's so much money being put into the human on a chip project that if you read about it, if you have any opportunity to work on these human mimicked organs, it's well worth your effort, because that will be that's our future. And for all of you in, what degree were you saying, biomedical engineering is a perfect spot to be at this point, is to have a degree in biomedical engineering. So that is an excellent future for anyone doing that. Conclusions, I think I covered everything already. I think I got all that. Just a few quick acknowledgements. Funding support for most of our work has been through the National Science Foundation. Right now it's through the National Institutes of Health. We have a company, 490 Biotech, that's investing some money in this research. And then we have Dr. Gary Saylor, who heads our center. Dr. Ting-Ting Zhu and Dr. Dan Close were the graduate students on this project, who now are postdoctoral students. They stuck around to continue working on this research. And then Pat Yeager, Dean Webb. Pat Yeager is one of our technicians. Dean Webb and Alexander Rogers are 2 of our undergraduate students. And our undergraduate students at our university when we get them into the lab, they do more work than most of our graduate students, and probably me as well. They do so much outstanding work as undergraduate students. So if there are any undergraduates around who can get into a lab, please try to get into a real research lab and do some real experiments, because it really helps. And if there are any questions,